All right, so we'll start the webinar. Good day to everyone. Good morning, good afternoon. So my name is Luka. I'm the CEO of Bio-ReCell, and I would like to welcome you all to our, first of many scientific webinars on the topic of next generation CAR T solutions. So Bio-ReCell, we're a commercial stage biotech company focused on cell therapy tools, reagents, and devices. And today, we're very proud to have in our group Dr. Anže Smole and Dr. Lenart Girandon, who will both have their talks on very interesting topics connected to CAR Ts. So just before we start, I would like to introduce both of them. So Assistant Professor Anže Smole leads together with Ilka Pohar the Immunology and Cellular Immunotherapy Group within the Department of Genetic Toxicology and Cancer Biology at the National Institute of Biology in Slovenia. He's also an Assistant Professor of Immunology at the University of Ljubljana. He earned his Ph.D. in biomedicine at the same university, and he did his postdoc at University of Pennsylvania. There he contributed to the development of different advanced CAR T therapies, and his research spans fundamental immunology and health disease, T-cell immunobiology, cancer immunotherapy, development of new therapeutic strategies, in which the immune system plays a central role, including, of course, cancer and autoimmune disease. And also Lenart Girandon, he's also part of the Bio-ReCell team. He has, decades-long experience now in cell therapy, spanning from stem cells, so MSCs, to CAR Ts, anywhere from development, clinical implementation, GMP manufacturing, and also right now he is deputy director here at Bio-ReCell, and he's basically managing all the R&D commercial applications here. So without further ado, I would like to ask Dr. Anže to basically present his talk titled Intrinsic Regulation of Cytokine Release Syndrome by Engineered CAR T Cells. Anže?
Okay. Hello, everyone. Luka, thank you so much for a very kind introduction. Thank you also for welcoming here at Bio-ReCell. It's an honor to be here today. And as I said, I come from National Institute of Biology, specifically, sorry, it doesn't move, I think. Is that? Okay. Specifically from Immunology and Cellular Immunotherapy Group that we established together with my dear colleague and friend Ilka Pohar. And we are not only a small group, but very proud and honored to be able to collaborate with young, passionate, and enthusiastic immunologists, some of which are on this photo, but not all, while some on this photo are now already our alumni, successfully leading their careers in biotech, pharma, or academia elsewhere. So just briefly, I'll outline what our group does. Basis for everything for us is basic immunology research, and then we are trying to bridge this fundamental immunology with clinical applications. And we're, of course, very interested in advanced immune cell immunoprofiling because we want to learn about how our immune system works in health and disease more in detail. And then we're also developing immune-based therapies for cancer and autoimmune diseases, and these efforts involve genetic engineering of immune cells and other things as well. Today, however, I'm gonna talk about work that I did while I was postdoc at the University of Pennsylvania School of Medicine, and this is just a disclosure related to today's talk. And as a little bit of background, what was inspiration for us, it really is Emily Whitehead. I'm sure that most of you heard of Emily Whitehead. She is the first pediatric patient to receive CAR T cells in 2012 at UPenn and CHOP. And in my mind, CAR T cells are now what they are because of this amazing young hero, Emily. And she had a acute lymphoblastic leukemia that was unresponsive to other treatments, and that's why they decided to pioneering CAR T cells. And as you may know, she is now cancer free, and that's sad till this where it is because of her, I think. So, but what is maybe less known about her situation is that when she received CAR T cells, very soon after that, she got very sick. She had difficulties with breathing, with blood pressure. She had a very high fever. And at that point, this was not predicted by preclinical models at that point. So it was a very serious situation for doctors, for researchers involved in her treatment. And I'll just say that these amazing geniuses, like doctors and researchers that were treating her, did many things, wanted to figure out what's happening. And one of the things that they did is they analyzed their blood for certain molecules to help them understand what's happening. And among these molecules, IL-6 was very high. And they said, as people with such broad knowledge and experience, they were able to identify and decide that if they use antibody that blocks IL-6, that could potentially help. And they indeed did that. This was really pioneering, brave and genius work, really, that they decided to do so. Her situation got way better very soon. And most importantly, she was cancer-free very soon after that, and remains so more than a decade later. So what happened now, we know that this was cytokine release syndrome, the toxicity that is now predicted and expected when doctors treat patients. And what's good about that is that antibody that was used, that's called tocilizumab, that is antibody that blocks IL-6 receptor, is co-approved together with CAR T cells. So doctors need to have that at site when treating with CAR T cells, so they can administer that if needed. And that, as said, was inspiration for us. And here we came up with the idea, why wouldn't we integrate into CAR T cells secretion of antibody based on tocilizumab, so that CAR T cells would autonomously treat cytokine release syndrome. And that's what I'm gonna tell you about today. And first, in order to do so, we developed a novel genetic platform, which we call UNIVECT. And first, I'll just say what the idea here was. This system enables inducible and temporal expression of desired molecule. And the rationale here is, like, when you get sick and let's say you take a medicine against fever, once you get better, you stop using this medicine, right? You wouldn't use it indefinitely. And here our rationale was similar. If we want to treat with some additional molecule, it's best if it's just temporarily there, as long as it's needed to provide therapeutic effect. And for that, we developed that novel genetic platform that enables expression of accessory molecule together with immune receptor, which in our case was a CAR receptor. And this accessory molecule is expressed in antigen-induced expression. We achieve that by rewiring endogenous T-cell activation signaling to the expression of this accessory molecule. So to simplify, once CAR T cell sees its target and recognizes tumor antigen, this signaling would turn on expression of this accessory molecule, which in our case was antibody that was able to block IL-6 signaling. Now, this sounds like a nice and reasonable idea, but there were several issues here and very many reasons why this would not work. And I would at this point say how it was important for me to have mentors that even though there are many things why this wouldn't work, they still supported that and just said, yeah, just go for it. Let's try that. So I think in science, sometimes you need to be brave. You need to risk things. Sometimes things don't turn out well, but sometimes you achieve something that has potential to really improve patients' lives. So that I think in our field is quite common here. So why I'm saying this, first, we didn't even know whether our inducible system would work when targeting hematologic malignancy. And here I'm showing you one I don't know that you see my cursor here, but we had these CAR T cells that were recognizing CD19 and inducibly expressed GFP. This was just a basic system, so that we wanted to validate how it works. And we have two target cell lines. One is CD20 negative and one is CD20 positive. And you see, when you co-culture these CAR T cells with CD20 negative cells, you see target cells showing up here. This is flow cytometry plot that shows targets here and CAR T cells here. And when they were stimulated or co-cultured with CD20 positive cells, you see that target cells are gone here. So CAR T cells indeed lysed cells through CD20, and on top of that, you see here expression of GFP, which you see is on y-axis. And that's exactly what we're hoping to see here, because GFP turns on once cells CAR T cells recognize their targets. And you see here, GFP is low. And keep in mind, this is log scale, right? That's how, so this is quite good ratio of activation. So that was great news for us that in principle that worked, and we moved forward with more questions. So first one was, can CAR T cells produce and secrete tocilizumab-based antibody? Single chain FVFC in this case. And keep in mind, at the time that we were working on these approaches, T cells have not been used to produce antibody-based molecules. B cells produce antibodies, and other cells, of course, in pharmacy, you know that you have cell lines that can very well produce antibodies. But CAR T cells, there were no reports that the T cell would be able to express such complex molecule and it would be biologically active. So it was first question for us. And the other question at that point was, will this molecule that blocks IL-6 signaling interfere with anti-tumor activity of CAR T cells? So that was, these studies took a long time to do. So at that point, it was so early that we didn't even know yet how blocking IL-6 affects CAR T cells also by using tocilizumab. So that when discussions were happening, also clinically relevant discussions, whether you can administer maybe tocilizumab preemptively or not, would that interfere with CAR T cells or not? Now we know that that's fine, that works. But at this point, we didn't yet know. And here, I'm showing you now another construct that the rest of my presentation will focus on. Here we now have CD19 targeting CAR T cells. So that's the target that's used in clinically approved CAR T cells. And now instead of GFP, we're now expressing this anti-IL-6 receptor single chain FVFC molecule. And here we are looking at lysis of target cells. We have three conditions. These are untransduced cells, just T cells alone, that you see they don't lyse target cells. Here we're showing lysis on y-axis, and here different ratios of effector to targets, and they don't lyse cells too much. And here in green, we have something that's very similar to now the conventional CAR T cells, and you see that lysis is quite efficient and increases when you have more effector cells. And at the same time, our construct that instead of GFP has now this tocilizumab also lyses target cells in a similar fashion. So at that point, we were happy that these cells were active and they were able to lyse cells, but we didn't yet know whether this molecule interferes with anti-tumor activity or not, simply because of that, because we didn't know whether tocilizumab is even biologically active, right? So if it was not biologically active, then it wouldn't probably have effect on lysis, but also wouldn't achieve our therapeutic purposes that I'm going to talk on next slide. So then we checked here also whether this molecule can at all be expressed in CAR T cells. And sure enough, we found that in non-stimulated cells, CAR T cells, you don't see expression, but if you stimulate with target cells or polyclonally with small molecules, you get expression of this antibody. And we were quite happy because at this point we knew that we can get T cells to express this molecule. But as said, we didn't yet know whether this is biologically active or not. And that brings us to our next slide. First question was, does this antibody that's supposed to block human IL-6 receptor alpha even binds to human IL-6 receptor alpha? And here we have target cell lines that express human IL-6 receptor. And to these cells, we added supernatants either from control CAR T cells that don't express ITOC, and we don't see any binding here. But when we add supernatant from cells that do express ITOC, we see binding happening here. And that was the first point that we saw, yes, that's something happening here. But that was not enough to advance further studies. We needed to know whether this molecule is biologically active. And I don't have time today to go in detail about this system, so I'll just tell you that we developed cell-based assay that enables us to monitor IL-6 signaling. And in this setting, we stimulated cells with IL-6 and monitor this IL-6 signaling. And you see when we added control supernatant, this here in black, you see that that's how IL-6 signaling is happening when we are adding more and more of this we are adding this control supernatant. So that's signaling that's turned on, right? Then when we add recombinant tocilizumab, you see if we add more and more recombinant tocilizumab, you see it increasing molar ratio. This signaling is dropping and quite drops to very low levels here. What I will also say is that you require quite a lot of tocilizumab to efficiently block IL-6. So that for us was an issue because we didn't know what's the capacity of CAR T cells, how much they can produce this recombinant molecule. But we saw here when we added supernatant that expressed this where anti-IL-6 receptor blocking antibody is expressed, we see that signaling is dropping as well. And at that point, we were quite happy to see that it's also biologically active, though it's less active than compared to recombinant tocilizumab as shown here. But that's in a way also expected. This is clinical grade molecule here that's isolated, and we here use supernatant from cells that express tocilizumab. So we're quite happy to see that. And with that, we advance to in vivo study. And I'll just tell you one thing. If you recall, initially I told you that these toxicities that they observed when they treated Emily were not predicted because they were not observed in preclinical mouse models. So that's important concept that not all things can be seen or transferred from preclinical models to then human data. But just in these times, amazing group from Italy for the first time developed mouse model, preclinical model that faithfully recapitulated cytokine release syndrome in CAR T cells. That's a very complex model that involves these mice in which human immune system is engrafted, basically. So they produce hematopoietic stem cells, and once they engraft, they would add cancer cells, this NALM6 cells, and once cancer is engrafted, we come in with 3 cellular products with untransduced T cells, with conventional CD19 targeting CAR T cells, and with CAR T cells that express inducible tocilizumab antibody. And now we are coming to our key slide. Here that's a little bit packed, but I do briefly through that. We wanted to know whether our innovative and conceptually new cellular product has biological activity in vivo. And this first graph here, we are looking at tumor growth. So again, this was done by our collaborators in Italy. And here we see days after CAR T cell treatment. And we're looking at untransduced cells, conventional CAR T cells, and CAR T cells in blue that should express ITOC. So tumor growth, when you add untransduced T cells, so T cells that are not engineered further to better target tumor cells, tumor outgrows. And that's why we don't see mice here, because they needed to be sacrificed or died here. And then when we see conventional CAR T cells in blue, you see that they nicely control tumor growth. And similarly, our construct with ITOC is also able to control tumor growth. So at this point, we knew that ITOC, at least in this model, doesn't interfere with tumor activity. But then we wanted to know whether it can affect cytokine release syndrome. Specifically, can it autonomously ameliorate cytokine release syndrome in vivo? And here we are looking on y-axis weight loss. That's one of key parameters that you monitor in this particular model, because when cytokine release syndrome happens, has such severe effect on mice that they lose weight, and substantially. You see, it drops really low to almost 70% of weight. And when we were basically constantly in contact with a group from Italy, for example, here on day 4, you see that control mice that have conventional CAR T cells engrafted, they're losing weight, but so were our ITOC cells. So we were very upset at this point. We thought that, unfortunately, this approach wouldn't work. So not the best days for us is here, but we continued these experiments, and then few days later, we received news from a group from Italy that these curves are starting to separate. And what happens here is you see that mice that have conventional CAR T cells, they have weight here, while ITOC group starts regaining weight. And this now translated in improved survival. We are looking now survival here over days, and you see in blue, all mice that were treated with ITOC CAR T cells survived, while more than half of mice that have just conventional CAR T cells implanted, they died out of cytokine release syndrome. And that's how this cytokine release syndrome is also serious in humans, right? If patients that start getting cytokine release syndrome are not properly treated with tocilizumab, for example, this is very serious. That could also lead to that in humans as well. So these data were really pioneering at that point, and we even didn't believe ourselves that this can work, because it was quite science fiction at that point. But so we were quite happy to see that. But we also didn't know exactly what's happening, what's mechanism, how these things work. We, for example, looked at what number of CAR T cells in blood, and what we noticed is that in ITOC group, at one point, cells start decreasing the numbers and frequency, while in control CAR T cell group, they remain at higher levels. So it has to do something also with the number of T cells. And then this will be my final data slide. We looked at cytokine levels in blood of mice, and we look at many cytokines that are associated with cytokine release syndrome. And you can see that each dot here represents individual mice that cytokines, most of them are lower in group that's treated with ITOC CAR T cells compared to group that's treated with conventional CAR T cells. Most of them, not all of them though, right? For example, IL-beta is quite similar. So at this point, we know much more about the mechanisms, what's happening, but we can conclude that this approach presents a conceptual advance of autonomous amelioration of cytokine release syndrome by CAR T cells. And even though further studies are necessary to now better understand the underlying mechanisms, what's really happening here, we can conclude that ITOC CAR T cells were able to reach the right balance of in vivo expansion, anti-tumor activity, and cytokine secretion to be effective in tumor clearance without causing lethal toxicity, without the need to administer any recombinant antibodies. So everything intrinsically built into CAR T cells. And with that, I have now most important slide here, acknowledgements. This work was done while in Powell Lab and the UPenn Center for Cellular Immunotherapies. This need to thank my mentor, Daniel Powell, and all members of the research group that substantially contributed to this work. Then we have our collaborators across Center for Cellular Immunotherapies, and of course this institute at San Raffaele from Italy that were willing to go with us together on this adventure that before we started, no one really knew what the outcome would be. So we are very thankful and honored that they were willing to collaborate with us. And of course, support from the University of Pennsylvania, Carl June, Joseph, John, Wary, and Sargil were all people who helped in this project. And of course, for all funding and resources. And I thank you for your attention.
Thank you, Anže. I really enjoyed the talk. I must say, you've done amazing work, and I could almost feel the excitement, the tension, the nerves when you were waiting for the results, the mice results. I mean, that's something that's very relatable, I believe, to every researcher out there. I think what you've done is very novel, interesting, and yeah, amazing results. So congratulations. We'll continue with the program. So we'll have our Q&A at the end of the session. So if you have any questions, please type them in. We'll continue straight forward to the next talk by Lenart. So Lenart is going to be talking about new paradigm in T-cell activation. So moving more from a basic science to applicative project, again, in the CAR-T space. So Lenart, you're welcome to start your presentation.
Yeah, thank you very much. Anže talked really a lot about how to build better CAR-Ts. I'm going to talk about, let's say, our contribution to make the production of CAR-Ts easier, cleaner, so that we would like to get them closer to the patient and faster to the patient. So it's a complementary talk, I would say. So I would just like to go briefly through CAR-T process. I know that the majority of people here know how they're being done, but the most important parts are like first purification of cells that we get out of patients. Then the reprogramming part was really nicely described by Anže. And then the next crucial step is activation and expansion of those cells. Albeit the expansion nowadays also is happening inside the patient. And at last, the administration. So here in the administration, we get to a point where the safety and efficiency of the product are measured, like because it's used in human beings. So for that, we need to first look into the purification and activation. And today's talk is mainly about activation. So why we chose to try to refine the work that has been done is because we can see here. So that was an experiment where we tried some nanomaterials that are used for T-cell activation, and try to figure out if they get internalized into the cell, because the majority of solutions use actually magnetic nanoscale beads, which are small. So this was, as you can see on the middle image, where the fluorescence photograph is presented. So here we can see that the beads that are outside of the cell are nicely fluorescent. However, we have stained them in such a way that once they get internalized, they stop being fluorescent. And on the far right side, you can see some of the beads that are actually noted with arrows, that we cannot see some of those, or the majority of the ones that are noted with arrows, we cannot see in fluorescent. That means that actually these materials are inside the cells. And as we know that the majority, when the cell has some reagent attached, that is directly implements how the risk management needs to go. So we figured out that maybe with size we can do some change. So we actually tried a lot of different materials. At the end, we went for a material that is like 5 times bigger than cells, which you can see on the left side in the above pictures, where the small dots are cells and the big dots are materials. So with this, we of course needed to figure out a way how to attach the antibody on. And again, our necessity or our decision was to to make it in covalent manner, so that we won't that they won't be able to be detached from the bead, so that actually we can remove it completely. And first, where we tried to, where we decided to use it is for T cell activation. T cell activation actually is enabling genetic modification and then proliferation down the road. We obviously, as everybody else, try to mimic the natural way of T cell activation with using of CD3 and CD28. That's the, let's say, we did nothing new here. However, what is new is that those antibodies, as I said, are covalently attached to the so big of a bit that it cannot be internalized. Since the material is so big and the cells are small, that means that we can easily remove after the activation of T cells. They can easily be removed just via strainer or some easy filter. Of course, we had to determine whether our solution has some potential in it. Did we do anything? Yeah, since we wanted to leave no residuals behind, we tested obviously if we can detect some antibodies. And on the below left figure, you can see that already after 48 hours and after 9 days, we're well below the lower limit of detection, even more so from a lower limit of quantification. And this is done with commercial, the best kits that we can get. And obviously also on the right hand side, you can see that staining with secondary antibodies to detect the antibody we know we added to our beads, we can see that there are no positives. So we got the direct proof that we are onto something, because we are not leaving any residuals, and that we can do something with this technology. But obviously, just no residues and no good results also mean nothing. So obviously, we need to show that the activation results are fine. So we here I'm presenting the work that has been done on activation. So during 48 hours, we can see. So we obviously were we wanted a comparison to the best kits out there, and we can see that the level of expression we were able to get was similar, but obviously together with the activation results, we need to look into the exhaustion markers, because if we overstimulate, overactivate the cells, that means that actually they can become old, they can become exhausted, which do not then clearly show some benefit in the patients. And here we can see that actually also our material, our reagent has the exhaustion markers not much different than the competition, which was also a really nice thing to discover, to see. And since we don't have our own CAR Ts, since we are more working with T cells before CAR T work, and because we have good connections, we collaborate in collaboration with National Institute of Chemistry. We did the comparison of the different activation reagents. And we can see on the left-hand side that our activation, our activation brings a little bit lower percentage of transduced cells than the rest. However, very interestingly, the killing assay is our reagent is the orange one. So on both figures. On the left-hand side, you can see that that the orange attract kit is a little bit lower than the rest. However, once we get to the killing assay on the right-hand side, we can see that the killing is bigger than in the rest, and we contribute that. I don't have the slide here, but we like attribute that to probably to the ratio we get. So because we tend to get more CD8 cells than CD4 cells, and we all know that CD8 cells are the ones that do the killing. However, yeah, so at this point we got to a point where our activation works, but is it scalable? Is it useful for somebody who is producing CAR Ts? So we then went into scalability here. It's also really a nice collaboration with the producers and with Scale Ready of some bioreactors. So we wanted to see if the technology is scalable. If we go to larger scale, does it activate the cells? Can we get better numbers of cells? So we went from like 96 wells to liter scale bioreactors. We tried to get the majority of them and test them, and we saw that it's scalable, that actually we can in bigger flasks we can get bigger numbers of cells out, which is also a nice thing. But as you can see, everything that is presented here is like open system. That means we need to the process is pretty tedious, because if you are to make this to be used in the patients, you need to use class A, class B room for in the GMP for the production, and this is quite costly and time consuming and also personnel consuming. That's why everybody is trying to figure out a way to be in a closed system, and we wanted to make that as well. So we developed also, or we're developing. So we made proof of concept, but we are developing this reagent also in a closed system way. And how we designed it is the majority of let's say GMP labs that are producing CAR-Ts have these welders or are using these sterile connectors. So since the majority has welders in their laboratories, we went into let's say setup that is easily connected to on the one side with the on the middle image. You can see that on the left hand side there is a bag with a sample, and then on the right hand side it's a bioreactor. So it's easily connectable both to the back and the connector. And here we really want to bring it closer to the production sites, where we know that the majority is using bioreactors, the majority is using some kind of way to connect them together. And this solution actually solved this. We tested also the efficiency of the activation, and it was also fine. This technology, this is just briefly. It's not about the activation anymore. This slide is more towards using the same technology for other purposes. What we are developing is also the cell separation. Besides, let's say, the activation, the cell separation is also crucial because we need to have clean cells in order to have the clean product at the end. But this is more, let's say, in development phase, and I hope to share more data on that at some other occasion. So the overview, how we tackled or how we see that the people like bringing reagents or devices to the market should go through is like first do this first research and development, like choosing the biocompatible material with favorable properties. Then develop functionalization. Here we also had to do a lot of different stuff to find the right one. Then obviously finding the antibodies that sufficiently activate the T cells. But then we had to confirm that the reagent is doing its task. Then obviously we had to learn how it behaves, what outcomes can it provide. And the last but not least were scalability and how the customer can use that in the production process. What we're doing right now is GMP compliant production and the QMS that will follow that. So we are in the final stages of this part as well, because it's crucial not to be. I mean, we want to be a support for academia, for academic endeavors, but also we want to support the real deal production of the countries. Thank you.
All right. Thank you, Lenart. Again, also a very nice talk about the activation reagent. So now that let's say the main part is over, we can continue towards the Q&A. And I'll just read. So I do have some questions here already from the viewers. So one of the questions for Lenart. So who in your mind is the competitor of the Attract kit? That is from Avinash Goswami.
Yeah, the majority of people actually is using Miltenys Transact. So actually, I think you were seeking this answer, but also Dynabits is pretty close. What we are like trying to be a little bit separated from is this soluble antibodies, because they're like they're there on top of the cell, like also in the final cell product. So it's a different class of the product, I would say.
And a follow-up question to that, how do you think you are better than Miltenyi or GeneScript?
Okay, so yeah, that's getting more into commercial part, but yeah. So definitely we are better in a way that that we can remove all the reagent can be removed out of the cells. The cells we our process finalizes is are without antibodies, without magnets, without any of these, and this then translates into less risk management that is needed for the regulatory.
So one question for Anže. So what is the next step in the research? So you've proven that that the cells can actually produce the IL-6, and where do you see that going into, let's say, as a product, or is there another version or another generation that you feel in your mind is the next step?
Thank you, Luka, for this very relevant question. One issue in the field of cellular immunotherapy and development of novel approaches is that there are so many brilliant teams around the world that did so much of brilliant new conceptual work published in top-tier journals with really strong data. But at one point, you need to decide which of these approaches and studies will be moved to clinical translation, and that's obviously very challenging question. So in that regard, my hope is that someone will see our work here with these CAR T cells and find it robust enough, relevant enough to maybe discussions about moving certain parts of concepts of that into clinics. But that's yeah, my hope at this point.
Do you see any other molecules being co-expressed next to IL-6 or any?
Oh yeah, that's another great question. Look at that, really addresses what we are trying to do. I think in our group, this is one part of this story that I chose to present in this limited time, but also in this paper, part of this was also this study's co-expression of IL-12 that was geared not towards improving safety, which ITOSI was geared towards, but IL-12 was geared towards improving efficacy in a safe manner. And we were able to show in preclinical models that CAR T cells upgraded with this inducible expression of IL-12 were able to control solid tumors in solid tumors models, whereas CAR T cells without this molecule were not able to do so. And one thing that's most interesting for me personally, and what our group also now really focuses on, that is basic immunology. So we also expressed transcription factors in these CAR T cells and showed improved intrinsic properties of CAR T cells, so that we would have better product that would maybe persist better or resist exhaustion or things of that sort. So
Thank you.
Thank you.
Just before Lenart, I do have a question again from the public, and I think I'll give this question to both of you because I think it really plays well into both topics. So, and I'd like you, Anže, to answer it first. So, is there any scientific proof connecting initial T cell activation or separation conditions with downstream CRS, especially when we're talking different ratios, etc.? What do you think?
So again, very relevant questions. if I understand this one correctly, if not, please come in with follow-up question, is that how you would predict performance of certain cellular products, how they will do in clinical application, how you can predict that just based off some in vitro ex vivo assays. So that's in a way holy grail in the field, right? How you would do that. So for cytokine and also for efficacy in cytokine B-cell, people are developing multiparametric assays when they score many different factors, so that they would try to describe as best as possible functionality of CAR T cells, for example, that would be predictive of their response in vivo. But that's certainly a challenge.
Mm-hmm.
Yeah.
Lenart, what's your opinion on that?
It's a good question, but it's a complex question. As Anže said, it's not easy to predict. However, I think, as I try to mention it at the beginning of my talk, I think activation is really one of crucial parts in the CAR T preparation, and it definitely affects how well the cells will be. So how effective, and also how long term they will be there. As I know, the cytokine release syndrome is there, like it's an acute part of the treatment, which means that the majority of things is just how efficient cells, CAR T cells, patient gets into the body. I think the activation can here have an effect, especially through the cells not being too activated. I'll say that.
Yeah, I mean, I'll comment as well here. I think I see, the activation part like priming the cells. You're basically setting them up for success or failure, right? So because my background is primarily microbiology, and if you compare that to infectious disease, some infectious disease, they use activate the immune system in a way that it can calm down. And then the others that overreact or suppress the immune system, like Ebola, in the end, basically your immune system is the one that kills you. So I think there's more to be learned here on that effect, but of course, for now, these are just more or less theories. Lenart, you also had another question, I believe.
It was for Anže. This I was really impressed by this mice work. I mean, this survival. I didn't see cytokine release syndrome such a big difference actually between life and death. I mean, it's like obvious that with your management of this cytokine release syndrome, the mice just survived, and the rest, even though they got CAR Ts, some of them died, or the almost half of them died. However, once now we're getting more towards the in vivo CAR Ts, and this kind of part. What do you think, would that approach also work or benefit in those that in the CAR T processes that do not take so much time outside the body, but take more time inside the body?
Lenart, thank you for this great question and for your positive opinion about our work. These results that we saw were quite striking. As said, we were hard time believing that in ourselves when we saw it. But keep in mind, this is still a model, right? A model, preclinical model in mice, as good as it gets, but there's no way of telling how that would perform in there. Because as we discussed now, I'm always amazed by the complexity of immune system, and the more you know how the many things are that you still have no clue about what's going on, and that's what drives still, right, in a way. But yeah, with this approach, conceptually, our hope was also that maybe cytokine release syndrome wouldn't even happen, right? That such product that self-sufficiently suppresses cytokine release syndrome would still result in tumor control, but would prevent of cytokine release syndrome to happening in autonomous manner. And now, when you go to this in vivo field, of course, yeah, that's definitely now the field that everyone is thinking about. This really profound and fantastic results coming out. I think approach like that would fit well in this system because it has all functions being built in, integrated, right? If you deliver that in vivo, you would still deliver CAR and this inducible dosage when cells gets this genetic material integrated or introduced in any way, it would be able to now sense and respond to, sense activation and response to activation by releasing antibodies. So it would be amenable, I believe.
Another last question. And this is more, again, bridging the gap between the basic science and, let's say, the end product. Do you think, or how do you think, with this approach by combining different molecules in the vector, affect the cost, or would the cost be more associated with the quality control aspects down the line? Do you see that like increasing the cost of the car like twofold, or let's say insignificant in a way?
Yeah, this is another great question. And as basic scientists, we focus mainly on things, how they work, how they make them work. But at one point, this economic is key, right? Also, we would like the therapies that are being developed to be broadly available. That's again back to in vivo CAR T-cells or off the shelf products, right? This is certainly hope that to get to everyone who needs that, right? Ideally. In specific question about our approaches, I didn't have time to go in detail about it, but now I'll say a few words about that. Clinical grade lentiviral vectors are expensive, right? And our approach that combines constitutive expression of immune receptor and inducible expression of accessory molecule was conceptually new as well. Because oftentimes people would use two vectors. One would deliver CAR, the other one would deliver inducible expression of accessory molecule. But with that, you have at least two issues. One is if you co-transduce with two lentiviral vectors, you would get non-homogeneous cellular product. In other words, one cell will receive CAR, but not inducible module. The other one will receive just inducible module and one part. And as a matter of fact, with these experiments, we know that 1/3 of cells would receive both, and only these are functional cells. That's potentially an issue for translation. You would like to have as defined as homogeneous product as possible. So this single vector system has this advantage here, and we were able to show that it works better than two component system in that regard. And on top of that, it would be beneficial in terms of cost of manufacturing. You need to create one clinical grade lentiviral vector versus two of them. But then more broadly about your question also, I believe that the more complex the system is, the better you need to understand that this comes with all additional assets and really trying to know what you're working with so it's robust and as safe and as efficient as possible.
Thanks. Yeah, I mean, that was basically my outtake from that is that I really like the fact that you didn't just combine different vectors and put them all in randomly, but basically to incorporate that. Basically, you need to do that once, and then you just manufacture the vector. But obviously, the quality control later down the line does take some time and effort. Well, I think, perfect. We're right on the hour. So with that, I would like to thank both presenters for taking the time, presenting the really amazing work. And of course, I would like to thank all of the participants that joined. The webinar will also be available online So follow us with the updates. We'll probably upload it in a couple of days. And with that, I would like to again thank you all and close the webinar. And till next time, so keep in mind we'll have more of these coming, different topics, and looking forward to seeing you next time. Thank you very much.